So, very good evening students, this is your Prasanna ma'am from NEET World. Here we are in discussion with the last session of Morphology of Flowering Plants. So far what did we dealt beta? Vegetative characters of morphology is done, floral characteristics are done, of which you will get a definite question from what? Floral characteristics. What all you have to cover from that beta? What all you have to cover from that? Floral characteristics from morphology of higher plants. Yesterday only I dealt, no? First, based on the flower symmetry, you have three types of flowers. What are they? Asymmetric, actinomorphic, zygomorphic. Based on the position of ovary with respect to other floral parts, you again have few types of flowers. What are they? Hypogynous, perigynous and epigynous. And then when it comes to androecium, gynoecium, calyx and corolla, two non-essential worlds and two essential worlds, of which androecium cohesion and adhesion with examples is important. Okay. And when it comes to sepals and petals, that is calyx and corolla, aestivation is very important. And lastly what? Position of ovules or arrangement of ovules inside the ovary, that is placentation. Amongst these 5 aspects you will get one question for sure every year. Okay. Now after floral characters, what is that we are going to deal with, ma? Yeah. Now seed, fruit and morphology families. Okay, seed, fruit and families. After this you come prepared tomorrow with the complete chapter of morphology. We will first finish off NEET PYQs tomorrow and then we will start anatomy of flowering plants. Okay. First you see seed. You have to know what are the parts of seed first. You have seeds. Seed mainly consists of 3 parts. One is seed coat, and then embryonal axis, embryonal axis at the apex it has what? Plumule, and below is radicle. From seed during germination beta, radicle is the first part that emerges out of the seed. Okay, first emerges out of the seed at the time of seed germination. Okay. And after embryonal axis, third is what? Cotyledons. Cotyledons are also called as embryonic leaves. Embryonic leaves. Okay. So seed consists of how many parts? 3 parts. First is what, Nana? Seed coat, embryonal axis. Embryonal axis at the apex region you have plumule, lower it is radicle. Radicle is the first part that emerges out of the seed during seed germination, after which you have what, Nana? Cotyledons. Cotyledons are also known as embryonic leaves. Okay, cotyledons are embryonic leaves. This will give the food, proper food or nutrition for seed germination. This basic thing is clear to you, know. So when you observe the structure of a dicot seed coat is made up of 2 layers. Outer layer is called as testa, inner layer is called as tegmen. Okay, dicot seed has 2 seed layers. Outer layer is what? Testa, inner layer is tegmen, and there is a small scar on the seed coat, which is called as hilum. Okay. So what is seed? Post fertilization, ovule is turning into seed. Yes or no? In ovule, let us once recall the parts of the ovule. Okay. Most of the angiosperms, yeah, most of the angiosperms will have what type of ovule, ma? Did you remember? Anatropous ovule. Okay, anatropous ovule is also called as what? Inverted ovule. And the stalk like structure is what? Funicle. Okay, small connecting link here between funicle and integuments is what? Hilum. Remembered? Okay, this hilum is small scar on the seed coat. Hilum will connect with the fruit. Fine. Ovary wall post fertilization turns to fruit. Ovule post fertilization turns to seed. Okay, ovule parts, the stalk of the ovule is funicle and the connecting part is hilum, integuments, and this is what? Chalazal end, this is micropylar end. Okay, hilum is hilum from the ovule. When it turns to seed, it is as a scar on the seed. Okay, this hilum is connecting with what? Fruit. Understood? Next is what? Micropylar end. Micropylar end is a small pore over which region ma? Small pore above the hilum. Okay, and this allows what? Water absorption during seed germination. Micropyle is the region which allows water absorption. Hilum is the region which connects with the fruit. Remember that. Next is what? This is the entry point for pollen tube also during fertilization. During fertilization, what is the connecting point, entry point? Micropyle is the entry point. Okay, like this only, no, pollen tube will come. Pollen pistil interaction we have learned, and then embryo. So here you have embryonal axis, right beta? This is the embryonal axis, if you can see. This is the embryonal axis. At the apex region it is terminated as plumule. Lower region is what? Radicle. Plumule and radicle is clear, right? Embryonal axis and embryo. This in most of the dicot seed, you know, after double fertilization, what is formed? After double fertilization, first male gamete, you have to recall this. Double fertilization, what happens ma? First male gamete will fuse with egg cell, forming what? Zygote. This zygote matures to embryo. What is the ploidy of zygote? Diploid. What is the ploidy of embryo? Diploid. Clear, right, Nana? Next, one more male gamete, yeah, it will fuse with secondary nucleus or polar nuclei, which is n+n condition you can take, or diploid, and this is haploid. This forms what? Primary endospermic nucleus, whose ploidy is
3N.
Triploid. This matures to endosperm. Remember, so in most nucellus are the cells which are present in the ovule. Don't get confused. Nucellus are the cells which are present in the ovule. Those are diploid cells. It has no connection with double fertilization. Remember. So one male gamete with a diploid zygote matures to diploid embryo. Another male gamete with polar nuclei or central cell forming primary endospermic nucleus, which by mitosis forms what? Endosperm. This is what? Triploid. Triploid endosperm. Right, ma? In most of the dicot seeds, what happens is this endosperm is completely consumed at the time of seed germination. In most of the dicot seeds. Very rarely, for example, castor, wherein the endosperm is still persistent in the seed. Okay, endosperm is not completely utilized at the time of seed germination or for the formation of embryo development. It is not completely utilized. Okay, so here this castor is having what? Endosperm. From that we will get that castor oil. Okay, so most of the dicot seeds are non-endospermic seeds or non-albuminous seeds because it is consumed by embryo development. Okay, only very few dicot seeds will have what? Very few dicot seeds will have endosperm. Those are albuminous seeds or endospermic seeds. Clear with it?
Ma'am, is coconut endospermic or non-endospermic?
Yes, that comes into different sector again. Okay, because it has cellular and non-cellular. Fine. It is endospermic. Okay. So here if you see, embryo has two cotyledons and an embryonal axis. Embryonal axis future root is what? Radicle. Plumule develops to future shoot. Next, two cotyledons, these are fleshy with reserve food material which provides proper nutrition and nourishment for the seed germination. And endosperm is usually absent in dicot seeds like pea, bean. They do not have any endosperm, but present in some dicots like castor. These are what? Endospermic seeds. Okay, endospermic seeds. Till here it is clear, ma? Any doubt anywhere with dicot seed? Easy, no? Yeah. Next we will look into the monocot seed. Yeah, monocotyledonous seed. Here, unlike the dicot seed, in dicot seed you have two layers of the seed coat, that is what? Outer testa and inner seed coat is tegmen. But here seed coat is fused with the fruit wall. There are no two layers of the seed coat. Here seed coat is fused with fruit wall. So this is fused. Best example is maize grain. Comes under what type of fruit? Caryopsis. Endosperm is persistent. Endosperm is not completely utilized for embryo development here. Okay, endosperm is persistent. This is large and food storing part of the seed. Okay, this is surrounded by protein rich layer called as aileron layer. One example I will take and tell you, you will understand it in more detail. Maize is the best example. You will get questions on this diagram also. In NEET 2024, you got a question on this diagram directly. Okay, so this is what is endosperm. It has two types of endosperm. Inner is what is starchy endosperm. Okay, starchy, which is surrounded by a proteinaceous rich layer. This proteinaceous rich layer is what? Aileron layer. Protein rich layer is aileron layer. And it has a papery cotyledon. This papery cotyledon is called as scutellum. This papery cotyledon doesn't have a reserve food material in it, but then it transfers the food material from the endosperm to embryonal axis. This embryonal axis at the tip region, it has what, Nana? Plumule. At the lower region it has what? Radicle. Only in the monocot seeds, remember, plumule has a covering sheet over it that is called as coleoptile, and radicle has a covering sheet over it which is called as coleorhiza. Understood? Any doubt here? So see, it has what? Most of the monocot seeds will have endosperm, starchy endosperm uncovered by a protein rich layer called as aileron layer, and it doesn't have fleshy cotyledons, it has a papery cotyledon. Just a second. Okay, let's get back, ma. So here with monocot seed, you have what? Starchy endosperm. Starchy endosperm is covered by what? Aileron layer. Okay, starchy endosperm with surrounded by protein rich layer called as aileron layer, and here papery scutellum is there. It is not providing nourishment directly. Okay, it is papery scutellum which is providing nutrition from the starchy endosperm. Clear, ma? So here it has a embryonal axis. This is driving nutrition from the endosperm to embryonal axis, which will give proper development for plumule growth and the radicle growth. Clear? Coleoptile and coleorhiza you will observe only in monocot seeds. Those are the covering sheets over plumule and radicle. Clear with the monocot seed structural description? Any doubt anywhere, beta? Clear, right? Yeah. Now for your identification, take this flow chart. A seed is of two types based on the number of cotyledons. Based on number of cotyledons. Firstly, tell me what are the parts of the seed?
Testa, Tegmen,
seed coat.
embryonical axis, Cotyledons.
Yeah, seed coat, embryo, cotyledons. Fine. Or else you can directly take seed coat and embryo. Embryo will be having cotyledons and embryonal axis. Fine. So here based on the number of cotyledons you have what, ma? Monocot seed. Yeah, dicot seed. If it is clear for you here only, this seed part, it will be easy for you in sexual reproduction of flowering plants. There are many connecting links with this and of that chapter in NCERT 12. Okay. So here monocot seed, most of them are what, beta? Endospermic. Endospermic or albuminous you can take. Albuminous seeds. Very few are non-endospermic or exalbuminous. Okay, dicot seed you see. Most of them are what? Non-endospermic or exalbuminous. very few are albuminous. Okay. So, here very few, for example, orchid seeds. Most of them are exalbuminous, very few, for example, castor. If this flow chart is in your mind with the examples, then it is enough. We are winding up very soon because you have physics class now. Okay. Fruit and morphology families we will deal in the next class. The seed part is clear to you know? Once it is done, let me know.